I think some of you have attended these events in person and know that they're sort of lively and interactive, and we're going to do our best to simulate some of that online. Today's event is about evolutionary dynamics, and we're going to cover a broad range of issues, ranging from real-world problems to abstract models. This is a field that's been incredibly exciting for the last 10, 15 years. Well, it's an old field, of course, 150 some odd years old, but it's been reinvigorated by new people, new ideas, new experiments, which mix together in really interesting, exciting ways, both going back to thinking about very fundamental questions about what's possible, but also trying to understand what's actually going on in the real world. And so it's a pleasure to have the day start with Richard Neher from University of Basel, who's going to talk, in particular, has made wonderful contributions to understanding how to bring those ideas about sort of the analysis of theoretical models together with real world data. I don't think any of us really knows for sure what's the best way to do these things. So what we're going to do today is we're going to ask that everyone stay muted, that you address your questions in the chat so everyone can see them. If something bubbles up and seems really urgent, I'll interrupt Richard. But otherwise, there'll be some natural pause points at which we can look at the chat and see what questions there are. Altogether, we have about an hour and a half, and I will stop there and ask Richard to start.
Yeah, hello everybody. It's my pleasure to contribute to this this series. I think 2 or 3 years ago I was actually in New York also contributing to this, so it's a pleasure to be again here and talk about evolutionary dynamics. And yeah, so let's just jump right into it. I'm curious how this works with the questions, so don't be shy, sort of ask and keep these questions coming. I'm hopeful that we'll have a good discussion. Let me sort of share my screen. So yeah, I'm also trained as a theoretical physicist, and I used to work a lot on abstract models of evolution and these kind of questions. And Ivana and Oscar will talk more about this in the rest of the day. But me in particular, over the past few years, I've been sort of digging deeper and deeper into the real world problems, meaning sort of actually trying to use these ideas to track, predict, interpret how these pathogens evolve. And in particular, this year, this whole operation has gone a little bit into overdrive with COVID-19, and there's been so fairly little time to think deeply about theoretical issues, but the real world issues have become all the more pressing. But I'll still talk a little bit about theory, but I'll start with flu, then do some theoretical discussion of theoretical problems, and then finish with sort of what we've learned about coronavirus, so SARS-CoV-2 and COVID from genomes over the last few months. Okay, let's start with flu. And there's actually many reasons to look closely at flu this year, because flu are the viruses where we have experience with pandemics, right? So in living memory, there have been 4 flu pandemics. And the one that is most well known is what's called the Spanish flu. There's no real good reason why it's called the Spanish flu. It probably started somewhere else. But as it so happened in 1918, there was a new strain, a new variant of influenza that caused an enormous pandemic with millions and millions of fatalities. And after that, after this pandemic, it sort of started to settle into a seasonal flu, like the one that we experience now. Every winter there is sort of a flu wave, and I'll talk more about that in a second. So this flu probably came from animal virus. It truly came from animals, probably from birds. And yes, sort of it caused this massive pandemic. It sort of started then to circulate as a seasonal virus over the years until the late 50s. In the late 50s, there was a second flu pandemic, which is sort of called the Asian flu, which was much less severe, but nevertheless, it was sort of sufficiently dominant that it drove the H1N1 flu to extinction. So, that then sort of circulated for a few years. Only for 10 years later, there was a new strain of flu, which caused another pandemic. And that is sort of a strain that, it's colloquially known as a Hong Kong flu, which is still around today and sort of keeps dominating our winter flu epidemics. Then for some somewhat mysterious reasons, this strain came back in the late 70s in a manner that was completely unchanged from when it disappeared. So that potentially was, we don't know for sure what happened, but it probably came out of a freezer somewhere, which sort of reseeded this sort of seasonal, every winter a new flu season, new flu outbreak until 2009. And 2009, some of you are, probably most of you are old enough to remember the swine flu pandemic 11 years ago. And the swine flu actually goes back to the Spanish flu from 1918. So they have a common ancestor. The 2009 swine flu and the Spanish flu have a common ancestor at some point in the early 20th century, which is one of the reasons why people were so scared of the Spanish flu 10 years ago, as the swine flu. But it turned out to be a very, very mild, flu strain, which nevertheless caused a pandemic, but it was a mild strain that just demonstrated to us how a pandemic unfolds without actually causing any problems. So as it happens, this 2009 swine flu finally sort of drove the Spanish flu descendants extinct. So we've now these two strains of influenza A, which are circulating as seasonal viruses. And if we look at this dynamics a little bit more closely, what I'm showing you here in this graph is the number of positive tests for the different kinds of flu. Here's in blue, influenza A, H1. So that is basically the descendant of the Spanish flu. Then you have here in orange, that is the swine flu. And in green, that is the H3N2 variant, which would be, this guy here. So then in addition, there's influenza B, about which I'm not going to talk about much. It's just it's a quite distinct, but a different kind of flu that's also circulating. And you see this very regular yearly pattern. So this is data from the US. Every winter, you have a spike, and sometimes the spike is dominated by the H3 variant. Sometimes it's dominated by the H1 variants. There's maybe a bit of an alternation, but most prominent feature here are sort of regular winter peaks, with one exception where you have this sort of weird sort of multi-peaked structure in 2009. That is the swine flu pandemic, and I'll talk a little bit more about that towards the end of the talk. But this is kind of the sort of disturbance that you'd expect when you inject a new pathogen that does compete with the other ones, but is basically hitting a mostly completely susceptible population. While these sort of ups and downs, these seasonal outbreaks, this is what you expect when you have a population where you just have an endemic virus, which most people have seen already in the previous years. Now, these are just sort of numbers. These are positive tests, very much like the SARS-CoV-2 case, the corona case numbers that you read in the newspaper now every morning, right? These are the number of positive tests. This is done routinely over decades already, that you test for the prevalence of these different flu viruses throughout the years. Now, what I want to do, I want to dig even a little deeper and use the genomes and the mutations in these flu viruses to track how they're spreading, how they're changing. And just so to illustrate the basic concept here. So we have a pathogen that's spreading, and each of these red dots here is an infection of somebody by a pathogen, right? Sometimes, the person recovers and doesn't infect anybody. Sometimes the person infects multiple people. And, the structure that emerges from this is that of a tree, right? So it's kind of vertical transmission of a pathogen, of which, some small fraction of individuals get sampled, right? Even with SARS-CoV-2, we don't expect that we sample every case. We sample a fraction of the cases, and of a fraction of these, we'll end up determining the genome, and then we'll find that there are particular mutations that happened somewhere on this transmission tree while the virus was replicating, and we can now use these mutations to reconstruct this transmission tree and sort of get some idea of how the virus has spread and changed. Now, in real life for flu, this looks more like this. This is a phylogenetic tree here of influenza H3, so that sort of Hong Kong flu variant over the past 30-something years. And the very early sequences here are on the left, down at the bottom, and the later sequences are on the top right. And along this tree, sort of, along each branch of these trees, there's sort of mutations that happen, very much like in this previous slides, right? There's mutations that happen on these branches as you go from left to right. And, these mutations, they, predominantly cluster in the globular head of a surface protein of the flu. So the flu is sort of this, it's an envelope virus, and there's sort of multiple surface proteins on it. And these proteins, they are subject to immune selection, right? That's what antibodies recognize. And if antibodies recognize it well, they neutralize the virus. So the flu is under constant pressure to change the antigenic properties of its surface proteins so that it can reinfect humans it has previously infected. And for the very same reason, the vaccines that we get, the flu shots, need to be updated quite frequently in order to keep up with the virus. And in fact, there's different gray crosses here that you see marked on the tree. These are vaccine strains that the WHO has been recommending over the past 20 years to be used as representative of the circulating population. And they get updated every year, every 2-3 years, depending on how rapidly the virus is changing. If you now look at a particular position of this protein, you'll see that over time, the amino acid at this position will change. And I have one such example down here, which is a particularly dynamic position, where it's a position 189, doesn't really matter for this purpose, but it sits somewhere up here on the protein. And over the past 35 years, this has changed its amino acid identity 4 times. So it used to be a lysine, then sort of an arginine was dominating for a few years. Then sort of, another mutation happened to a serine and so forth. And you see these transitions, and this is a particularly clear example here, where in about a year or two, the entire population of flu goes from being dominated by one variant at this position to another. So these are sort of, prototypic selective sweeps, if you want, where the virus population changes in response to immune selection. So one remarkable feature that you might have noticed is this tree that I just showed you. It's basically just one long trunk, right? Without any long side branches. This doesn't need to be this way. You could also expect a situation like this, where the tree at some point split into two lineages, and this happened with the influenza B. I just wanted to point this out here. This is in itself a very interesting research question. I wasn't going to talk about it at greater length, but the fact that some flu strains seem to do this very linear, sort of low diversity but rapid evolution, while others sort of are in this, others seem to be more prone to splitting into multiple variants, which then sort of circulate independently of each other, is sort of a very, very interesting question. Okay, any questions at this point that we should answer, Bill?
No, I think, okay. Here's one. Since you paused right at the lineage split, do we know what determines that, or?
Yeah, so our idea here is that this is going to be some interaction between sort of how far, so these different lineages compete, right? And when they compete, they drive each other extinct. But if they manage to evolve away from each other far enough that the competition diminishes, then they actually accelerate. So it's a bit of a nucleation scenario where you sort of need to get these two lineages or two different variants sort of far enough away from each other that they...
Sorry, and here you mean far...
In some sort of antigenic sense, right? In some, they compete for the same individuals to infect, right? And if two very similar strains infect a particular, or if one strain infects a particular individual, a very similar strain can't infect that same individual because they are, from an antibody perspective, too similar, right? So they sort of need to be able to evolve away from each other enough, despite this constant competition.
And does geography play a role here too? I mean, could they just go to different parts of the world and...
Yeah, so geographic separation certainly helps in this, right? Because it provides it reduces competition between lineages. But flu has been a pretty good jet setter here. So it's quite remarkable how well mixed flu is across the globe. And it is true that H3N2 is actually more mixed than the influenza Bs, because influenza Bs, at least some lineages, tend to infect kids a lot, and kids don't travel quite as much. So that might be contributing to this problem. Yeah.
Okay, and there was a question, this sort of classic question: why does it peak in the winter?
Yeah. So let's postpone this, because I'll come back to this in the context of SARS. Okay.
And there's a kind of variant. What is it that makes the lineage split any different from the regular variation?
I think it's chance.
Okay.
It's a nucleation scenario. It sometimes happens, sometimes it doesn't, and then the rate at which it happened might differ between different strains.
Very good. Okay.
Okay, very good. So what for flu, we've now for several decades have sort of a quite remarkable system in place where there's hundreds of labs on the planet that constantly collect flu samples. And they then either sequence them themselves or they send them to a small number of centers to be sequenced. And so we have a quite, at least until recently, unprecedented sense of what viruses are circulating where and to what degree. Right now with SARS-CoV-2, we actually have an even bigger data set and even sort of denser sampling. But up until sort of beginning of this year, this was sort of quite unique in how well this how this virus is being surveyed. This is sort of organized by sort of the global influenza surveillance and response system, which it's a quite remarkable sort of global operation where sort of every major country contributes to, and it's also often extremely fast. So the data that we find in that database GISAID is often, a sequence could show up there just 2-3 weeks after it has been sampled. So somebody's sick, a swab gets taken, and 2 or 3 weeks later, we have that sequence publicly available. Which, in peak months of a flu winter has been, it's hundreds of sequences a week, right? And that sort of very rapidly exceeded the capabilities of these different centers that sort of do this flu surveillance to analyze and interpret these data. And this is sort of something that me and a colleague, sort of Trevor Bedford in Seattle, sort of tried to tackle a few years ago. So we came up with this website, NextFlu, which was meant to provide exactly that. And I'll sort of try to, do this live here. So what you see here, and it might be a little bit too small for you to see everything. I'll try to walk you through a bit. So what you see here is some sort of a dashboard with a phylogenetic tree on the left, a map of where these strains come from on the right, and sort of a display here of which positions in the genome. This is the HA segment, which has like 2 different proteins and a signal peptide. These are the positions where there is diversity, and these bars are essentially showing the entropy at these positions, right? And normally, we update this every week, and normally you'd have data there that is just 2 weeks old, but this year is different. So we have essentially not seen flu since March. It has been more or less completely suppressed globally. The only recent sequences we have here are, interestingly, it's a monastery in Cambodia where they had an H3N2 flu outbreak, but that's kind of the only flu that we've seen since May, which we'll see whether this holds also in the coming winter. But it's sort of quite a striking difference to what we've seen previously. Normally, the most recent strains here would just be a few weeks ago, but currently you see this very sharp line with a few outliers, which is basically the beginning of the lockdown, the stay-at-home orders across the world, which have pretty much put an end to flu circulation. It is still circulating somewhere, but in much, much, much lower numbers that it's basically under the radar. But what I'm showing you this just as an example of how we are using these sort of modern sort of technologies to basically go away from this. You have a bunch of data, you analyze it for a few months, you write a paper, you send it to review, a year later you get it back. So we're trying to cut this entire process short to make sure that we can actually use these genetic information in a very timely manner to make sort of public health decisions, right? And this is sort of an interactive display. You can zoom into different things. You can look at what every virus here. That's a virus from Maryland from sort of 7th of March and so forth, which has a bunch of other characteristica. So yeah, so this is just to give you a sense that when one can sort of take this entire analysis of genomes here to sort of a new level, to actually make it sort of available to people at the front lines that are trying to make these decisions about what goes into the vaccine and so forth, in a much more timely and informative, interpretable manner. Good. So yeah, that's just a screenshot. And so the one thing, it's easier to operate with screenshots than live. So the one thing that I'd like to point out here is that this is currently sort of colored by geographic region. So there's North America, there's South America, Europe, Africa, Asia, South Asia, and so forth. And what you see that the tree here is mostly salt and pepper, right? So there is some regions where there's more America, some regions where there's more Europe, but pretty much every variant is found everywhere, which is sort of testament of the fact that we used to be flying around the globe quite a lot. And it'll be very interesting to see how over the next couple of months, if flu comes back, what kind of a geographic separation and demixing we've seen on this, given the fact that intercontinental travel has been dropping by one or two orders of magnitude. Good. So after this sort of bit of a 30,000-foot view of flu, I'd like to go into a little bit of theory and ask whether we can. This was mostly about tracking, sort of analyzing the data as quickly as we can and sort of disseminating the results to the decision makers. What can we do beyond that? Sort of can we predict, right? Sort of instead of just cataloging what happened, can we also project what happened in a meaningful way? But is there sort of a question that we should address at this point? Otherwise, I'll just go on.
No, I think, let's see. There was a question about the winter peaking of different strains. Are they overlapping with each other, or are they competing?
So that goes back to this guy.
Yes.
Yeah, so it's interesting. There seems to be somewhat of a mutual exclusion of the H3 and the H1 variants. So you either get one peak or two. Also, this is a log scale, right? So, but the bees are different. The bees sort of happen pretty much every year, and they're often like 2-3 weeks later. So the bees tend to peak in February, whereas the A's tend to peak in January. It's a little hard to see here. So here, for example, is quite clear. Here it's quite clear. This is also lumping data together from all sorts of, the US is pretty big. So in order to answer that, you would want to dig into different regions of the US. But yeah, so there is some competition with the Bs being slightly shifted later in the year, but there is almost an exclusion between the H1s and the H3s.
Good. Okay. Yeah, I think you can go on.
Good. So can we predict? And the main idea is the following. So this goes back now to evolutionary models, right? We are trying to put sort of mathematical descriptions of this dynamics that we are seeing globally. We clearly see there is a lot of dynamics, right? The different strains interact, there's sort of mutations that spread. So there is sort of competitive dynamics and sort of a selection for variants that can infect a bigger slice of the human population. And these mathematical models, they tend to be pretty simple if things happen one at a time, right? If there's one mutation which arises, spreads, nothing happens for a while, and then the next mutation comes. So you have a pretty easy mathematical description. Predictability, however, is pretty low because at some point the next mutation will happen, but you don't really know when. It's sort of all kind of waiting times, exponential waiting times, and so forth. Now, as you add more mutations, if more things happening at the same time, these models tend to get kind of complicated, right? Because you're now sort of dealing with a many body problem, and you have different mutations that sort of compete for the same thing and things like that. But sort of our idea here is that, once you actually have a large number of mutations, once you have a diverse population, and this is just a little sketch here of each black bar being a genome, these dots being mutations, and obviously I didn't want to draw like 100, but imagine you have a large number of these different mutations, all of which have sort of kind of a similar size contribution to the growth rate, to the replication success of the virus. What you then have, or you expect, is sort of a pretty Gaussian, sort of bell-shaped fitness distribution in the population. And that's a tautology, right? Sort of fitness, the ones with high fitness, they'll grow faster, they grow up, and the ones with low fitness go down. And as a result, you have these entire waves, these fitness waves sort of translate to higher fitness. Of course, stuff doesn't get sort of indefinitely fitter. Instead, sort of the population is changing too. There's immunity that's building up and so forth. And these two processes, they balance each other out. So you essentially have sort of a fitness distribution which objectively stays in the same place, but is sort of turning over in the sense that the individuals at the very tip of the wave, they grow and kind of drift backwards with the population catching up. And these ones at the end, at the low fitness tail of the waves, they just go away. And we'll probably hear a little bit more about that in the theory and in the other talks later today. So, while this is sort of a fairly intuitive picture, it turns out that describing this mathematically is pretty tricky because, all the important bits happen at the very, very tip of this wave, where despite the fact that we're dealing with a large population and many mutations, and you'd hope that everything will become sort of nice and sort of averaged and smooth again in some limit of large numbers, out here in the tail is sort of a very rare events, are outsizedly important because they exponentially amplify and have, huge downstream effects. Nevertheless, it turns out that, pretty much all models of this sort end up having a fairly universal limit, and that is what I want to briefly sketch and then show you how we can use these ideas to try to predict. So what I'm showing here in this slide is sort of a comparison of coalescent trees of sort of the trees that emerge as these things replicate for two different scenarios. And here, apologies for the different orientation. So down here at the bottom, this is the present. These are the leaves of the tree. At the top, that's the root of the tree, obviously, right? Sort of the root at the top, leaves at the bottom. And sort of as we go from up the slide, that's going back in time, right? And on the left, you have just an example of a tree that would come from a neutral process, where there's no real competition between the individuals. It's just that stochastically, one individual has more offspring than the other. And if you play this forward, you will end up with a tree structure where everything goes back to one individual eventually. But most of these merging, this coming together of lineages, happens very quickly until you are in a position where you only have two, three lineages left, which typically sort of splits the population pretty even into groups of kind of families, if you want, of similar size. Now on the right, you have the corresponding tree for a population where you have a lot of this fitness variation and sort of a rapidly moving wave and strong competition between the individuals. And you see that the structure is quite different. First of all, there is sort of a long time at the bottom where nothing really is happening. All these lineages go back and don't merge with anything. Then you have, deep in the tree, these kind of events where many lineages come together very quickly, which over here you only observed at the very tips. And then you see that there's a very skewed pattern of branching where you often have a minority of the individuals descend from one branch and the great majority from the other branch. Right? And this is happening here in a nested fashion, sort of multiple times. And it turns out that this structure here is actually converging to a mathematical coalescent ensemble that's called the Bolthausen-Snittman coalescent. And the details of this are sort of a little bit more complicated. I just want to give you sort of a little bit of an intuition as to why we see these features. And one is that when you sample your individuals, you're most likely sampling from somewhere in the middle of this fitness distribution, right? And as you then sort of trace their lineages back in time, so you go to their parents and grandparents, great-grandparents, and so forth, all these lineages end up converging to the very high fitness end of the distribution, right? That is because conditional on having offspring, you end up with more fit individuals, right? Because everybody has parents, but not everybody has kids. So parents, by conditioning on looking at parents or great-grandparents, they will end up being from the part of the population that reproduce more. Right? So this is the first part of this dynamics. You track these lineages from the bulk, from the center of the fitness distribution to the tip. And because there's many individuals in the center, not much is happening in terms of lineages meeting, right? In order for lineages to meet, you have to deal with a small population. In a big population, it's very difficult for lineages to meet. Now, once we are all here in the tip, that's when the real competition between different lineages happens. And sometimes one of these lineages is sort of lucky enough to just jump forward a little bit through some lucky mutations. It can then sort of grow transiently faster than everybody else, generate sort of some exponentially growing clone, which has a size distribution with a very, very long tail, a power law tail. And at some point, the wave sort of catches up, but it's these sort of transiently fit individuals that generate these bursts in the tree, because they, for a short time, can sort of claim a they can expand for a short time, and through this transient exponential expansion, sort of claim a macroscopic chunk of all future offsprings. And so this is pretty much all I wanted to say about this coalescent structure. But the insight, the take home here, what I wanted to make for the next few slides, is that there's bursts in the tree which correspond sort of to transiently high fitness individuals. And yeah, so these bursts in the tree are things like this, which correspond to somebody just getting ahead of the game here for a short time. Okay, some more questions?
No, there was a little exchange about where the logarithms go, but
yeah.
But that seems to have resolved itself. It is.
Yeah, others have more patience with lots of little logs.
Yes, it is. It is interesting that, right? it's harder to sweep through a larger population, but on the other hand, you have more individuals mutating, and so anyhow.
Yeah, so it's lots of
seems to have been
sort of
resolved
competing.
by the audience itself, which usually
Excellent.
is not polite. Usually not polite to do that in parallel with the speaker, but in this case,
It's the benefits of virtual.
right. So please.
Good. So yeah. So and I want to use this insight, this correspondence between these high fitness individuals and these sort of transient bursts in the tree to ask the question whether we can actually use this to estimate fitness of the leaves and thereby sort of predict what variants are going to dominate the future and potentially pick sort of a close relative of the future, which would be interesting from a sort of influenza vaccine design point of view, right? So you'd like to be able to anticipate which flu strain is going to dominate the future. And the basic setup that we are talking about here is the following. So you have a present, right? And you'd like to say something about the future. But of course, nobody tells you anything about the future, nor do you know explicitly what fitness these individuals have. So I have arranged them here on a fitness axis, right? But that, of course, is unknown to us if we only know the sequence. And the backbone of the tree, which eventually is going to connect to the future, that is always going to be here in the very high fitness tail. So, we'd like to pick this individual, right? Because it is sort of the closest relative of the future. But what we're asking here is, so is there any information in this tree structure that we can use to kind of rank the leaves here by fitness? And now it turns out there is. So we've sort of, and the details of this are a little bit complicated, but you can calculate, making some approximations on sort of who competes with whom, and using some sort of branching process approximations when you would really have to sort of account for a bunch of global constraints. But making these approximations, which are sort of valid on sort of short enough timescales, you can come up with a scheme where for each node of the tree, you can associate, you can calculate the posterior distribution of fitness given the structure, both the topology and the length of the branches of the tree. And essentially this works by kind of associating with each branch an object which sort of tells you how likely is it that I have a certain fitness here at this point and a certain fitness here at this point, given I observe offspring of this branch, given that there is something sampled that's downstream of this. And this has sort of, there's sort of two competing things here. Of course, the higher the fitness of this object, the more likely you are to find offspring. But at the same time, if the fitness on this branch is very, very high, you're very unlikely to see a long stretch without any other branching, right? So the fact that on this branch here, that there is no branching kind of limits the fitness of this thing from above. And so these are the competing tendencies that go into this fitness propagators, which you eventually will have to solve numerically. It's sort of sort of a little PDE that you have to solve, and you can then sort of using this sort of somewhat independence assumptions, use transfer matrix sort of recursive methods to calculate the fitness posterior on every tip of the tree.
So Richard, maybe yeah. I'll interrupt here with a question, which is about the definition of fitness and what's maybe on what are the timescales on which one is doing this. So fitness changes because, and so fitness is a growth rate, and it changes because it's assigned to a genome, but of course there's also the immune system of the hosts.
Yeah.
So how do I think about this? Is there an approximation in which the immune systems are rapidly adapting or something, or?
So in this specific calculation, what goes in here is, so we're assuming, yes, there is we're assuming the total population size is constant, which essentially means that we compare everybody's growth rate to the average growth rate of the population. Right, so we kind of subtract the average growth rate from everybody's growth rate. And then if you grow faster than, if you have a above average growth rate, you're expanding. If you have below average growth rate, you're contracting. And sort of what keeps this balance between the increasing mean fitness and the adaptation of the individual lineages, that is kind of implicitly the coevolution of immune system and the pathogen population. But yeah, that is the crutch that most of these models use, that you assume the population size is constant, which you impose. And Oscar might have some interesting things to say about this later, because he has ways of putting in different constraints. Yeah, you impose that the population size is constant, and achieves this by fiat, setting the mean growth rate to 0.
Okay. And there was also a question, which maybe, I don't know if you want to answer it now, which is whether there's a kind of Goldilocks zone for population sizes that somehow is balancing between the adaptability of the population and your ability to infer the structure of the tree.
Yeah, I'm not sure I can answer that in a meaningful way. So the ability to resolve the structure of the tree depends mostly on how many mutations are there. So that what's the mutation rate in particular? Is there sort of a large number of neutral mutations which just provide information about the structure? While sort of the population size is indeed an important characteristic, because in very, very large populations, this is, yeah, so you need to be in some sort of a transient, not yet asymptotic regime for this to work, simply because, yeah, otherwise, if you're in a super large population, you've basically forgotten much of the fitness by the time you get to the leaves. But yeah, it's a bit of a subtle issue, and I probably just would be mumbling confusingly if I went on.
Okay, so maybe we'll leave it aside.
Anyway, sort of what sort of comes out of this somewhat abstract mathematical object here is a fairly intuitive interpretation that structures like this here, where you have one lineage which rapidly splits into many, this corresponds to high fitness, as we already sort of kind of knew going into this, right? So in order to observe a structure like this, we have this thing to rapidly divide multiple times and leave offspring in the present. And it turns out that at least at close to the leaves, this is the fitness of this object is monotonic in just the total downstream tree length. So the number of branches that descend of it multiplied by their length. And this we turned, so because this is kind of a pain to actually calculate in practice, and it also makes a lot of somewhat specific model assumptions about how these mutations accumulate, that they're all very tiny and infinitesimal. Turns out there is a fairly useful and simple heuristic that captures most of this information. And that is what we call the local branching index. So if you have a tree like this, which is a rooted tree, so this is the root, right? You can, for each point in this tree, calculate sort of a weighted tree lengths. For each point in the tree, you can calculate the tree lengths in the vicinity. Right, and I illustrate this here. So you start with this rooted tree, you just unroot it, and say we were looking at this point here. This would be this corresponds to this point here. You then sort of put sort of an exponential kernel onto that point and just integrate the total tree lengths in that vicinity with this exponential weighing. So this index is going to be always high when you're at a point that splits in many things, right? And it's going to be kind of low for points like this, right? Because there is not a lot of tree volume surrounding this point, or there's a lot more tree volume surrounding this point. So it's basically just a measure of local bushiness, and that turns out to capture pretty much all the fancy things that we calculated on the previous page. I just wanted to show you a little bit how this is working out in simulations. So we can just do a simple numerical experiment here. We simulate evolution forward in time. We sample the sequences. We reconstruct trees from these sequences. We infer the fitness and then compare to what we got, right? And if you do that, you actually see that what you get out of the simulation is just sort of a long file with sort of binary sequences 0, 0, 1, 1, 1, 1, 1, 0, 0, 0, and so forth, right? But it turns out that the information in them there is information on fitness. So here I'm sort of correlating the inferred fitness with the true fitness, just as an example. And here we are sort of quantifying this correlation coefficient as a function of the mutation rate, which is essentially the ability of us to resolve the tree, and also the degree to which the model that we're assuming, meaning sort of small infinitesimal changes in fitness, how that is matched up in the simulations. And you see that, yes, it's sort of happily increasing these predictability of fitness, so the ability to infer fitness from a static snapshot. And that is important, right? So the insight here is that you can take individuals all from the same time point, sequence their genome, and make a statement about the dynamics. You can make a statement about which individuals are likely to increase and dominate their population and which ones are on their way out. And of course, this dynamics, it comes from the tree, right? All these individuals are connected by a tree, so that tree is providing history in a way, which allows you to turn a snapshot into a time derivative, if you want. Good. How does this work for actually picking a good ancestor, right? So for flu, our main interest is to actually find a good representative, and here it's actually working even better than the sort of prediction of fitness. And how we quantify this here is the following. So we have these two populations, right? And we'd like to find an individual here, which is a good representative of the future. And we will compare this to just sort of the random distance, so the average distance between these two, and the optimal distance. And then specifically, so what you see in this graph here on the y-axis is the distance of our pick, sort of the individual that ended up having the highest predicted fitness. On the y-axis, you see that distance normalized by this distance. So 1 is basically a random pick. And on the x-axis, you see the distance of the post hoc closest, again, normalized by the average distance on the x-axis here. So if you're on the diagonal, meaning your prediction is as good as the best thing that could have happened, right? And if you're above this dashed line, that means you're doing worse than random. But in a good fraction of cases in these simulations, you end up with a pick that's close to optimal, and there is some fraction in between where it's not bad, but it's not great. And quite rarely, in a few percent of the cases, you actually pick something that's worse than random. So after this successful simulations, we obviously asked to what extent does this actually hold for real flu. And the result is the following here. It's a similar quantification. It's a few years old now, but it kind of worked the same way since. Whereas this line at 1, that's kind of the random threshold, right? So if you're above this, you're doing worse than random. If you're on this line down here, then you're essentially picking the optimal. And in many years, yes, you end up picking the optimal. In some years, you're sort of middling. And here, early in '95, there's hardly any data. But yeah, it seems to be picking out useful representatives of the future population. And interestingly, it really doesn't care about the fact that this is flu, right? So there's no influenza-specific input, which bothered us in the beginning, because at the same time, sort of Marta Luksha and Michael Lessig, they sort of published with a similar goal, sort of a very different approach, where they were sort of trying to rather explicitly account for all sorts of flu properties, and they end up having similar prediction of similar quality, and we've since sort of tried to combine them in various ways, and there's actually not that much, by combining these different ways, there's not that much to be gained, and that is still slightly puzzling. But maybe I actually have some insight why this is so hard. But maybe before we get there, are there questions?
Oh, the chat has been quiet in this part.
The chat has been quiet. So because what I'm going to do now, so I showed you that this is working very well in theory, and it not only picks the good ancestors, but it's on simulations. It also has a very significant correlation with fitness. So there's something to it which works.
Sorry, saying that the chat was quiet, of course, produced a question.
Okay.
So at what mutation rate does predictability level off? And does it eventually decrease because if you had too many mutants?
Yeah, so if you had too many, so it depends a little bit on how you scale mutation rate with effect size. So you would, yeah, I don't have sort of a good, this is arbitrary units here, and I don't have a good dimensionless number for you to, on the top of my head for you to answer this question. That would require a few minutes of thought on my end. So I don't really have an answer.
Okay, fair.
But okay. Yeah, what I was saying that, this clearly sort of picks up fitness out of a static snapshot in a population that is close to the model that we are using to derive our fitness predictors, right? And in flu, we can't do this experiment, right? Because we don't actually know the true fitness, right? The only thing that we can do for flu is do this comparison, sort of how well are we doing in terms of how well are we finding good representatives of the future. And this is not actually testing the fitness inference at a very accurate or specific level, right? And so, postdoc Pierre Barin in the lab, we set out to actually sort of try to understand a little better, sort of what is it actually that makes flu predictable. And it turns out there's sort of very, very serious limitations to the predictability of flu, which we hadn't previously anticipated to that degree. And the first thing that Pierre looked at is if you take any kind of any trajectory of a mutation, and I showed you a few trajectories early, which did these very nice sweeps, sort of coming up without any interruption, going to 100% and fixing, right? And I told you back then that this is a bit of a cherry-picked example, but it turns out this is actually fairly atypical. So if you look at these frequency trajectories and condition on these frequency trajectories of going through a window of frequency of say 30%, and ask sort of where do they go afterwards, right? Are they persisting? Are they continue to rise, or are they essentially sort of are they turning around, or are they sort of just randomly meandering? So if you do this, you find the following picture. So the majority of trajectories here, they start low. There's a few which do a little excursion here because they kind of jump through the window, but removing them doesn't make much of a difference. So you have all these trajectories, they come up, they go through this window here because that's how we condition them. And afterwards, some fix, most of them go away again, and the average is actually essentially flat. So there's fairly little information on future dynamics if only you condition on a recent rise, which puzzled us because you'd expect that if there is any kind of sweep-like behavior left, if that is typical to some degree, you should see that this average is sort of continuing to creep up. And you see that in other viruses, but you don't seem to see it in H3N2 influenza. So furthermore, if you actually look sort of, if that mutation initially was conditioned to be in a frequency interval of around 30%. And if you ask sort of where is this later, this is a bit of a wiggly funny lines are a little weird, so we should have just made it a histogram. But you see, like 30 days later, you have sort of a bump which is kind of centered around this, maybe slightly higher. 60 days, it's sort of spreading out. 120 days, and after about a year, you have essentially a flat distribution with already quite some contribution that fixed, and you've essentially lost all information from the beginning, with fixation happening at rates left and right that are essentially given by the frequency at the beginning. If you are at 30%, 70% will disappear, 30% will go to frequency 100%. So there's very little information in the future frequency that is sort of carrying it. So we are conditioning on rising, and that conditioning has pretty much no information on the future, which is certainly something that sort of puzzled us. So we dug a little deeper and then asked sort of why on earth do we seem to be able to predict flu with things like the LBI? Now it sort of turns out that you can make a very simple predictor, which is basically just taking the consensus sequence, like at every position of the sequence, you take the majority amino acid and use that as the predictor of the future. You can show that this is actually the optimal sequence representative in a neutral model, where the only thing that happens is sort of random fluctuations. And it turns out is the strain with the highest local branching index is most of the time pretty much the same as the consensus sequence. So much of the predictability for flu, we believe, comes from a tendency of the local branching index to pick consensus-like sequences, which is of course a little disappointing because we were hoping that we're actually picking up a genuine element of fitness here, as is suggested by the numerical simulation. But I have to confess that I'm less confident that flu can be meaningfully predicted from genetic sequence data alone. Because the dynamics of the human immune system and so forth, all these different environmental factors seem to be randomizing this dynamics to a degree that it doesn't look like a neutral population dynamics, very clearly not, but it's also not something that seems to conform with our ideas of sort of traveling wave models. Anyway, so this is sort of the flu bit, and I now wanted to turn on to COVID, but this would again be a good place where we just can take a short pause for questions.
So there was a general question about why predictability is possible, but maybe it was good to wait a moment because you.
Yeah. So in the theoretical, in the model that we solve numerically, predictability is possible. So there is a way to actually get fitness, so an estimate of growth rate, a meaningful estimate of growth rate from the sequences alone, and that is sort of what I was stressing earlier, right? That you have a snapshot of sequences all coming from the same time, and you would naively think that there's no dynamical information in there that would allow you to you want fitness is essentially a derivative of frequency, right? So in the snapshot there is a priori there's nothing to calculate the derivative from, right? But the derivative comes from the tree, right? So the tree groups these sequences into close relatives and not so close relatives, and it also tells you how how close they are related, meaning at how far ago their common ancestor was. And that is sort of where the time information comes from that allows you to pick glean some information about the rate of change.
So the other question that came up was that it sounds like the models that you're thinking about are sort of independent site by site, and there's a question about
no. So the model that we use to derive that theory is an architectural model where the fitness of each individual is determined by all its mutations at the same time, and they're completely linked. While sort of here for the neutral model, the consensus being the predictor of the future, there it actually doesn't matter. It's in the neutral case, everything just factorizes in some way.
I think the question was about functional interactions, epistasis.
Oh, I see. Our model doesn't care, right? So it's just jumps in fitness, right? Whether they are due to epistasis or not, there's no recombination here. And in that sense, as long as the distribution of fitness effects stays constant, it doesn't matter whether they're epistatic or not.
Right. You just need that weak form of independence where it's not like there are some mutations that open up a huge spectrum of possibilities and others that close it down.
Exactly. You need sort of that the distribution of possibilities stays the same. That it's kind of in a statistical sense, your spectrum of mutations remains invariant.
Good. And just to be, I think this is a good paraphrase of one of the questions. I should think about when prediction is possible, of course you're predicting a distribution. I mean, or somehow fundamentally the prediction is of a distribution. And so there's some assumption that what you're seeing is being drawn fairly out of that distribution, which is...
Yeah, so it's sort of a statement about whether the set of sequences we are operating on is sort of an independent and representative sample of the circulating viruses.
Which is a
strong assumption, and we sort of try
something like ergodicity, right?
Yeah, so we're trying to sort of sample like from every place on the planet, from every month, a bunch of viruses, right? But yeah, no, there's big sampling biases associated with this, and there's sort of white spots on that map, which all make these measures. And it's just surprising that they still work, right?
And then there was one more question, and we should let you go on, which is the neutral broadening of the condition time series diffusive or faster or slower? So if you think about projecting forward.
So the broadening of what exactly?
Let's see how quickly I can find somebody. Oscar, yes.
Can you hear me? Yeah. Well, Richard, I asked you that question, I guess, via email before. So you see these time series which are conditioned, and then on average they stay constant. Yeah. But so then it's a broadening process. So is that diffusive, faster or slower?
I think it's faster than diffusive. But the challenge we have here that it's overlaid by this annual cycle. Right, and that's where then sort of also these frequencies are not exactly equilibrated across the globe, and as the northern hemisphere enters into the winter, there's a whole bunch of things that rise, and then the southern hemisphere. So there is on top of these frequencies in any given locality, there is this sort of global mixing of different processes which have sort of strong constructions and contractions and expansions. But I would expect that this has long tails, precisely because of all these reasons. So I don't think it's diffusive.
Okay, thank you.
But good. Yeah, no, I know I owe you a response on that email, or Pierre rather. Anyway, so good. What I wanted to do in the last 20 something minutes or so is get a little bit into SARS, and Now, even if you're half as sick of it as I am, that's probably it's probably already enough. But still, since it's dominating our sort of daily lives, it's useful to. I hope it's interesting to share a few thoughts on it. And since it's it seems like, at least to me, like a century ago now, that at the very beginning of this year or the end of last year, there was this news and somewhat like the third, fourth, fifth page of your newspaper on this mysterious virus outbreak in Wuhan, which nobody knew anything about, and probably it's just sort of a bunch of people that got infected at this livestock market, but who knows. But then sort of obviously stuff started to develop very quickly. After this first initial pneumonia cases at this, connected to this fish market, it's actually been a remarkable ride. Only a few days later, Chinese scientists had sequenced the entire genome of this famously unknown virus and established that it's a cousin of the SARS virus from like 17, 18 years ago, which of course made everybody pretty scared because SARS back then had an enormous fatality rate. And which was somewhat more surprising is that just a few days later, we found cases of this virus in Thailand, in Japan, then sort of end of January in Washington State, and then sort of all over Europe. So by end of January, we found sort of cases of this in many places on the planet, right? And by that time, it was pretty much clear that there's probably biggish outbreaks underway somewhere which are not yet identified, and that's certainly what happened in Italy. Now, what are we actually looking at in terms of what kind of virus are we dealing with here? So SARS-CoV-2 is a beta coronavirus, of which there are multiple different ones which circulated or have circulated in humans. So there is SARS-1, which caused these outbreaks in 2004, 2003. There's MERS, which you might have heard about. This is a Middle Eastern respiratory syndrome virus, which is something that circulates in camels, and over the last decade or so was repeatedly found in people that interact with camels, but there is no sort of sustained transmission in humans. And then there's two more, which are actually quite interesting. And these are regular common cold viruses, which circulate in humans. They come back every winter, and some fraction of the colds you have in winter are due to these guys. So it's clear these beta coronaviruses are viruses that are very capable of circulating in humans, and sometimes they are very severe, sometimes they are mild. So if we zoom in a little more into this clade of SARS-like viruses, so we have SARS-1 here and SARS-2 here in red, and in blue there's all sorts of viruses that are isolated from different animals, mostly bats, but also these pangolins. There was a lot of talk about pangolins back in the beginning of the pandemic. We still don't know still don't have a clear origin of this. This closest virus here, the bat RATG13 virus, sampled in 2013, is about 3-4% divergent at the genomic level from SARS-CoV-2, which translates into a common ancestor sort of maybe 30, 40, 50 years ago. So it is clearly not the immediate ancestor of this, and we don't really know what happened in between. What I want to discuss now for a few minutes is what happens within SARS-CoV-2. You see here these red dots, they're basically all in the same place, meaning they are very, very similar, at least compared to the diversity of the other SARS-like viruses. And I want to zoom into that now a little bit. And unsurprisingly, we are doing this analysis on NextStrains, the platform that my lab's running together with Trevor's lab. And we essentially update a phylogenetic analysis of SARS-CoV-2 every weekday. And we've been doing this since the beginning of the pandemic, sometimes even more often. So, but yeah, if you want to get an up-to-date snapshot of this, you should go to the NextStrain website. But just to recap a little bit, sort of how this developed. At the very beginning, the first thing that you notice, this is January 26th. At that point, we already had some 20-30 virus genomes available. And what you see is that you don't see much, right? So this is not much of a tree in a way. So here at the very left, you have kind of this prototypical original Wuhan strain. And then sort of as you go to the right, you have viruses that are 1 mutation away from this, or 2 mutations, 3, and there's one guy here that has 5 mutations away. But for all practical purposes, these genomes are more or less identical. And which sort of made it quite clear at the beginning that, you know, this is not something that spilled over from animals multiple times, but there was one singular event, some single spillover from animals to humans, which then spread and caused a pandemic. So that was sort of the first insight that these sort of comparative genomics, these phylogenetics would give us. Since these first sort of 20-something genomes, things have been really somewhat blowing up. We now have about 130,000 full genomes of this virus, which is actually a substantial fraction of all known cases, right? I think we have, whatever, a couple of tens of millions of cases these days, right? So there's, you know, we've sequenced about 1% or so of them, which, you know, is quite remarkable. And I don't think has ever been the case before for a respiratory virus. The other thing that you see in this graph is that much of that sequence deposition and you know, public availability is very fast. So let me explain you that graph sort of a little bit. So each of these magenta dots here is the collection date of a sequence, which is associated with some date here. And the other dot, the turquoise dot onto the right of it, is the date when this sequence was made publicly available. And, you know, quite often these dots are only, you know, 1, 2, 3 weeks apart. Of course, there's always also backfill, which people uploading in August sequences from March, but, you know, there is a substantial amount of sequence available within 2 or 3 weeks of sampling, which gives us a more or less real-time view of how this virus is spreading across the globe. And it has since sort of diversified into sort of multiple different variants that we at NextStrain here have categorized into these 5 clades. 19A and B are sort of the variants that originally circulated in Asia. And then sort of 20ABC are the variants that sort of ended up dominating Europe and the Americas. And you see that, you know, nowadays, most of these viruses are between sort of 10 and 15 mutations away from the original strain. These mutations, they accumulate, you know, somewhat gradually and predictably. So what you see on this graph here is the number of mutations a virus has accumulated as a function of time. And you see that's a, you know, pretty, you know, it's noisy, but, you know, fairly linear sort of, you know, predictable increase with about sort of 20 something mutations a year. So that means every 2-3 weeks you get a mutation. They don't happen, you know, perfectly randomly. Sometimes you get 3 at a time. Sometimes for 8 weeks you get none. But you know, there is a sense that you have a kind of molecular clock that the number of mutations tells you something about how long this virus has been circulating. Okay, so that is the, you know, well, an overview from like a few weeks ago. I'll sort of hop over to the website to give you. Oh, I don't have this open. I meant to preload this. But yeah, here you see sort of a geographic distribution that, you know, the blue ones, 19A, 19B, are the ones that dominate Asia, while the rest of the world is sort of dominated by these later clades, 20A, 20B, and oh, it's still loading. I'm not sure why this is so slow.
Richard, while it's loading, maybe you could say a few words about the role of decreased cost of sequencing and everything, and whether that evolution of technology is going to make big changes in the future, or whether we've really felt so this is all possible because of those changes, but what's the projection?
Yeah, so yeah, sequencing has become much more ubiquitous and also much easier and sort of less less sort of capital intensive, with especially these sort of small sequencing devices like nanopore. I think what we'll see with these developments is yeah, them being used more and more also for diagnostic purposes, for sort of differential diagnostic metagenomics and these sort of things. You know, in terms of tracking a disease like this, I think we've almost maxed it out. So some countries like the UK are sequencing. You know, the UK alone has sequenced 100,000 viruses. So half of all available viruses come from the UK. If every country was doing that, we would basically, yeah, I don't think we would probably have pretty much every variant represented at sort of a high enough level to do sort of some serious analysis. Yeah, I think the next thing we'll see here is sort of this being used more and more in for diagnostic purposes. Anyway, so this has loaded now. And this is sort of the most recent analysis, where you see here, for example, there's you know, some viruses from Singapore that were sampled on the 29th of September. So just about 10-ish days ago. Here's sort of a similar one from Wales. So we have, you know, for many of these clusters, we have sort of a, you know, a very sort of tight rapid turnaround. This cluster here, for example, is this New Zealand outbreak that was in the news a few weeks ago, that sort of triggered the renewed sort of shutdown in New Zealand. They have basically sequenced every single case of this and have a you know, pretty good handle on it now, and sort of shuts this down. So I invite you sort of to, you know, play with this. I'm not going to sort of go through all the different things, but I've sort of prepared a few snapshots here that just give you some sense of what this can be used for. So there's one site which you might have heard about in the news, and that is sort of the position at 614, position 614, the spike protein, which, you know, has made sort of headlines multiple times as sort of being, you know, the virus mutated and now it's sort of way worse than before, which is not true. We've seen these kind of bogus sort of alarmist stories over and over again, but it's still interesting to see that, you know, you had a variant which emerged at some point in January, probably still in Asia, and then sort of within a few months sort of displaced most of the otherwise existing variants. And this variant also, it managed to spread globally before all these travel restrictions were in place. So we see now variants spreading in Europe or in the US and so forth separately, but before, you know, in February, March, they were sort of spreading globally and sort of and before any of the travel restrictions kicked in. But this variant in particular has now been studied, you know, quite intensively, and what it looks like is that it does increase transmissibility of the virus by some degree, which probably helped it in this sort of rise to dominance. But it is still not perfectly clear whether this transmission advantage was sort of the decisive effect, because the dispersal of this variant has also been very strongly driven by founder effects and chance. So these variants happen to have seeded the big outbreak in Italy, which then seeded the rest of Europe, which then in turn seeded the outbreak on the east coast of the US. And sort of by going through sort of multiple of these sort of amplifying big outbreaks, it could also have just risen to this prominence even without any selective effect. So one has to be quite careful sort of in over-interpreting the dynamics like this. But what NextTrain does allow us to do is sort of keep a close eye on all these variants in real time, and sort of try to find mutations that, you know, potentially, you know, affect binding of the virus to human receptors, that might interfere with vaccine development and things like that. So it's important from a sort of public health perspective to keep a close eye on this. And this is what we are being kept pretty busy with at the moment, because sequencing continues to go. So different regions now develop different variants. So there's a lot to stay on top of. But one of the aspects in which this genomic sequencing is also very useful at the local level, not only at this global which variants are dominating, is that it allows you to complement contact tracing. Right, and of course that's only sort of relevant if you are in a situation where you can actually trace. If you have sort of big community spread and thousands of cases every day, contact tracing is sort of a mute cause. But in places like New Zealand and Singapore and sort of other places that sort of try to keep the virus circulation at very low level, this kind of molecular epidemiology, sort of using genomes to reconstruct transmission, can actually help focus the resources that you have for contact tracing quite a bit. And the idea is the following: so as there's the same tree that I showed you before, there's mutations that happen, and you have a bunch of things that get sampled, right? So what you end up then getting are the genomes with these mutations marked here in these diamonds, which allow you to reconstruct this tree, which then sort of tells you that, okay, the cases A and B, they have plausibly been, they could be part of the same transmission chain, while cases D, they're part of a different transmission chain. So there's no point for me sort of trying to look for sort of a single source for A, B, and D, because that would have been ages away, and I have no point of finding them. So you can sort of focus your resources. Basically what the sequences allow you is you have one outbreak in a city, but you can sort of partition this outbreak into multiple independent outbreaks that you can investigate in isolation on the basis of the genomic similarity. And that means that if you manage to do this sequencing with a very fast turnaround, the genomic epidemiology can help you elucidate processes that happen on timescales of 2 weeks or more, while the contact tracing, asking people, who did you go to dinner with last night, that sort of helps you to resolve the structure on shorter timescales. So it's essentially sort of a timescale complement for the work by the public health authorities. Just sort of one little example here from Switzerland. So Switzerland, my colleague Tanja Stadler here at the ETH, she is putting a lot of effort into sequencing. And what you see here, for example, in Switzerland, this is only highlighting sequences from Switzerland. You see sort of multiple sort of distinct clusters, which all sort of go back to whatever was circulating in March, but which allow us now to basically investigate different outbreaks separately from each other, and also learn more about the transmission patterns between different demographics, different geographic locations, and things like that. There's sort of one example here where we have a cluster that emerged over the summer, which is actually seen in multiple places in Europe, which suggests that this might have been a cluster that actually lives in a holiday location where sort of many people went over the summer holidays and brought it back to sort of various places across Europe. All right, so I wanted to just spend a few minutes on on sort of what I think we've learned, and where I think this is going, and what we need to be very alert about. Unless there's any questions now, I would just go through this and
maybe
take questions at the end.
There is a question, but maybe we can hold it till you finish. And
okay, so what did we learn over the past months? So we certainly learned that social distancing or physical distancing is very effective, right? So we have in lots of places around the world, we have suppressed outbreaks. And we've also learned that, at least over the summer, many Asian and some European countries have reopened without catastrophe, right? But some opened too much and and then saw sort of exponential increase in cases again. What has frustrated me is that most decision makers don't seem to realize that case-based interventions like test, trace, isolate, and quarantine are much more effective and much less disruptive if you have low numbers, right? That you'd like to use these sort of case-based interventions to keep the epidemic in a subcritical domain where you don't see exponential growth, right? But the political constellations seem to have been pretty much everywhere such that you can't justify sort of measures in absence of healthcare distress, which is sort of has been sort of one of the most frustrating experiences over this pandemic. But one thing, and I think this slide is from a few weeks ago, and by now maybe I should remove this question mark. This pandemic sort of hit the northern hemisphere in late winter, early spring. And by the time we implemented most of the physical distancing measures that we had, also the weather sort of turned nice. And over the summer, it seemed, at least in much of Europe, that it's fairly easy to suppress this virus. But we are now heading into winter. And I think what we are seeing across Europe now with sort of fairly rapid rises, but also in the Midwest of the US, Wisconsin, Dakotas, and things like that, we see sort of rapid rises in cases where it seemed like it wasn't much of a problem in the summer months. So, and this comes back to the question, why seasonality for flu? And I think they have basically the same, why winter peaks for flu? I think they have basically the same reason. And one big one is the behavioral component, right? In temperate climates, you spend a lot more time indoors in winter than in summer, right? While I might sort of go with friends to the park and have a barbecue in summer, this is not an option in winter. Instead, you go to a crowded bar or a restaurant, things like that. There's also sort of an environmental component that, windows are closed, the indoor air is drier because of heating, which means that aerosols stay suspended in the air for longer and so forth. So there's all sorts of reasons why we think seasonality has an effect. People have argued that it's probably not so important for a pandemic virus because a pandemic virus can also spread in summer. There is a large population of susceptible individuals. But that doesn't sort of negate this point, even though this argument is often being made, just because the fact that a virus can also spread in summer doesn't mean it can spread even better in winter. So, this brings us back to this graph here, which I showed at the beginning, right? These sort of winter peaks of flu. And I want to look at this peak a little closer, right? Because this is a situation that is fairly similar to what we had now, only that this virus happens to have been sort of 100 times less serious than SARS-CoV-2, luckily. So this virus emerged in Central America at some point in early 2009, and started to spread in larger numbers in March, April 2009. And what you see is that it kind of disrupted the normal flu circulation patterns, right? It sort of started a peak here, basically when flu was supposed to be over, and then sort of started a very large early peak the following year, and followed by a void. So let's zoom into this a little bit. So this is the 2009 H1N1 flu in the US. And I've broken the US down here into, there's sort of various healthcare regions defined for the US. And I've broken them down in southern and northern ones. And what you see what happened in the northern US, sort of a kind of an early summer peak, followed by low, school holidays and all of this, and a second big peak in October, November. The South had a different trajectory. So it never saw this huge peak that you had in the North. It sort of had a slow burn over the summer, followed by yet another peak, and then generally sort of a less modulated course. And there's very clear parallels here to what we see with SARS-CoV-2 at the moment, right? So the South has basically had SARS-CoV-2 throughout the summer. It didn't have an early spring wave. And we'll see what happens now as we go into fall. One has to say influenza is it's probably more driven by kids, so the schools will have played a bigger role as they do for SARS, but the overall environmental patterns are probably pretty similar. In the UK, we've seen a very similar pattern to the northern US. So a summer peak, an early summer peak, a summer low, and an early winter peak again. Other European countries like Germany, they kind of skipped this first peak. They had a very minor peak, but then a very quiet summer, followed by a big peak in fall. And to me, and this is actually data now from the 1918 flu, which also came in a small wave in the beginning and then a second winter wave. So, for all these reasons, I think there's good reason to expect that the next couple of months are going to be very, very difficult in terms of SARS-CoV-2, which is sort of reinforced by the notion that, these viruses here, so these are the beta coronaviruses again, and these are these human common cold viruses, which we studied actually back in February of this year to get sort of a bit of a blueprint of what seasonality might look like for SARS. And if you look at them, you'll see that they have a very pronounced seasonality as well. So the seasonality is not something that's unique to flu, but if you look at the incidence of these seasonal coronaviruses, you see sort of a very, very regular pattern with peaks in early winter, which when you sort of take the whole thing modulo 1 year, you see here for the different viruses, especially the HKU1 OC43, that they sort of tend to pick up in October, November with the peak around New Year. And I think all of this, even though we don't have direct evidence for any sort of seasonality of SARS-CoV-2 at present, I think there's sort of multiple lines of evidence that point towards quite a significant seasonal forcing, which will make the next couple of months pretty tough. So that's pretty much all I wanted to say, with just one last remark. So what should we be expecting sort of going forward multiple years? It could very well be that this virus is going to turn into just another cold virus with sort of its summer peaks. So if that, this simple model where we assumed some immunological memory of a few years, you would get now sort of a period of a few years after the pandemic, which probably is over by the end of next year or something like that. You'd get a few years of very low circulation before this sort of starts to get into some pattern of seasonal oscillation with oscillating peaks in the southern and the northern hemisphere. Good. So that brings me to the acknowledgement. Sort of the work on theory and flu was mostly driven by these gentlemen here. Sort of Boris Schreiman, sort of my longtime collaborator, Trevor Bedford, with whom most of the next strain work has been done. Oscar, with whom we worked on the Bolthausen-Slittman business. Colin was involved in the flu prediction work, and Pierre, he was the one who analyzed these limits of predictability that I showed you. And then I have an extra acknowledgement slide here for next strain, because there's lots of people that are involved in next strain, and it's been a terrific collaboration with Trevor since 2014. With all sorts of people involved, and especially sort of James, Emma, Ivan, Moira, and Tom have been the drivers behind keeping next strain for SARS-CoV-2 up to date, which has really been sort of a tremendous effort in maintaining and updating this every day. Okay, with this, I thank you for your attention. I'm happy to take further questions, and stick around for a little.
Great, Richard, thank you.